Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Continuing Education, SOCE, Master's Degree Programs, Master of Science, Food Nutrition, MSCDFSM, Revised First Year MFN 002 Nutritional Biochemistry Unit-7 Lipid Metabolism 7.1 Introduction. Lipids are a heterogeneous group of organic compounds. The major dietary lipids for humans are animal and plant triacylglycerols, sterols and membrane phospholipids. You may recall reading about their structure and properties earlier in Unit 2. Here, in this unit, we shall focus on their metabolism. The process of lipid metabolism involves synthesis and degradation of the lipid stores. It also involves the production of the structural and functional lipids characteristic of individual tissues. This unit gives an overview of lipid metabolism at the cellular level, which would provide enough background for understanding the aberrations with regard to lipid metabolism and its related diseases. The first part of the unit, Section 7.2 Lipid Metabolism I, focuses on fatty acid metabolism, i.e. issues, reactions related to degradation and the synthesis of fatty acids saturated, unsaturated, in our body. The second part of the unit, Section 7.3 Lipid Metabolism II, looks at the metabolism of neutral fats, phospholipids, cholesterol etc. In addition, you will also find information regarding hyperlipoproteinemias, ketosis. What do these terms mean? Read and find out in this unit. Objectives. After studying this unit, you will be able to explain how fatty acids are oxidized for the production of energy, describe the synthesis of fatty acids, discuss the metabolism of triacylglycerols, phospholipids and cholesterol, relate the cholesterol and lipoprotein metabolism to hyperlipidemia, and discuss the significance of eicosanoids in human nutrition. 7.2 Lipid Metabolism. You learned earlier in Unit 5 that the lipids are absorbed through the intestine. As these molecules are oils, solubilization, emulsification of dietary lipids is accomplished via bile salts that are synthesized in the liver and secreted from the gallbladder. The emulsified fats are then degraded by pancreatic lipases. These enzymes, secreted into the intestine from the pancreas, generate free fatty acids and a mixture of mono and diacylglycerols from dietary triacylglycerols. Following absorption of the products of pancreatic lipase by the intestinal mucosal cells, the resynthesis of triacylglycerols occurs. The triacylglycerols are then solubilized in lipoprotein complexes, complexes of lipid and protein, called chylomicrons. Triacylglycerols synthesized in the liver are packaged into VLDLs and released into the blood directly. Chylomicrons from the intestine are then released into the blood via the lymphatic system for delivery to the various tissues for storage or production of energy through oxidation. The triacylglycerol components of VLDLs and chylomicrons are hydrolyzed to free fatty acids and glycerol in the capillaries of adipose tissue and skeletal muscle. Finally, what is the fate of fatty acid? Let us find out. 7.2.1 Oxidation of fatty acids. Fatty acids released from chylomicrons and VLDL are transferred across cell membranes by passive diffusion, which depends upon the concentration gradient. Their oxidation consists of a. Activation, b. Transport into the mitochondrial matrix, and c. Reactions of beta oxidation. Let us next learn about these three stages. A. Activation of fatty acids. Fatty acids cannot undergo oxidation in the form in which they are normally present in the body. They must be converted into a form which can be catabolized. This process is called activation, and fatty acid is converted into an active intermediate. Fatty acids must be activated in the cytoplasm before being oxidized in the mitochondria. This activation is catalyzed by fatty acyl-CoA synthetase. At least 3 acyl-CoA synthetases, each specific for a particular size, length of fatty acid, are known. Let us see what are these. 1. Acetyl-CoA synthetase. This acts on acetate and low molecular weight fatty acids. 2. Medium-chain acyl-CoA synthetase. This acts on fatty acids with 4 to 11 carbon atoms. And 3. Acyl-CoA synthetase. This acts on fatty acids with 6 to 20 carbon atoms. The activity of acetyl-CoA synthetase in muscles is restricted to the mitochondrial matrix. The medium-chain acyl-CoA synthetase occurs only in liver mitochondria, where medium-chain fatty acids, obtained from digestion of dietary triacylglycerols and transported by the portal blood, are metabolized. Acyl-CoA synthetase, the major activating enzyme, occurs on the outer mitochondrial membrane surface and in endoplasmic reticulum. Activation of fatty acids results in the formation of an ester between the carboxyl COOH group of the fatty acid and SH group of the coenzyme A, forming fatty acid ester of coenzyme A. The reaction favors the formation of fatty acyl-CoA, as the pyrophosphate formed is hydrolyzed by the enzyme pyrophosphatase, PP1 plus H2O2P1. Thus, activation of a fatty acid molecule requires expenditure of two high-energy phosphate bonds. B. Transport of acyl-CoA to mitochondrial matrix. You learnt above that the activation of fatty acids takes place largely in the cell cytoplasm. However, the enzyme required for oxidation is present in the mitochondria. The activated long-chain fatty acids, therefore, must be transported into the mitochondria. However, the mitochondrial membrane is impermeable, not allowing passage to fatty acyl-CoA ester. Hence, the transport of acyl-CoA is achieved by carnitine, gamma-beta-hydroxy-gamma-trimethylumonium butyrate. Two carnitine acyl transferase are involved in acyl-CoA transport: carnitine palmitoyl transferase I, captain I, located on the outer surface of the inner mitochondrial membrane, and carnitine palmitoyl transferase II, captain II, located on the inner surface. The activated long-chain fatty acyl-CoA ester reacts with carnitine. In the presence of carnitine palmitoyl transferase I, CoA is released and the fatty acid forms complex with carnitine called acyl-carnitine. This is able to penetrate the inner mitochondrial membrane and gain access to beta-oxidation systems of enzymes. Here, another enzyme, carnitine ACYL carnitine translocase, acts as an inner membrane exchange transporter. Thus, ACYL carnitine is transported in, coupled with the transport out of one molecule of carnitine, as shown in figure 7.1. This carnitine is generated when ACYL carnitine reacts with CoA to form once again fatty acyl-CoA, catalyzed by carnitine palmitoyl transferase II, located on the inside of the mitochondrial membrane. Once inside the mitochondrion, the fatty acyl-CoA is a substrate for the beta-oxidation machinery, as discussed in the next step. The role of carnitine in transport of fatty acids into the mitochondrial matrix is given below in figure 7.1. Figure 7.1, carnitine cycle C, beta oxidation. The major pathway for fatty acid oxidation is beta oxidation. The process of fatty acid oxidation is termed as beta oxidation since it occurs through the sequential removal of two carbon units, as acetyl-CoA, by oxidation at the beta carbon position between alpha 2 and beta 3 carbon atoms of the fatty acyl-CoA molecule. The reactions take place entirely in mitochondrial matrix. Oxidation of a saturated acyl-CoA with an even number of C atom to acetyl-CoA requires a repeated, sequential action of the following four enzymes, and the steps are highlighted herewith and in figure 7.2. 1. Acyl-CoA dehydrogenase E, enoyl-CoA hydratase E, beta-hydroxy-acyl-CoA dehydrogenase 4, acetyl-CoA acyltransferase, beta-ketothiolase or thiolase. 1. Acyl-CoA dehydrogenates acyl-CoA, i.e. removes 2H+, at the alpha and beta C atoms. This causes unsaturation to give alpha, beta unsaturated acyl-CoA, or increment 2 unsaturated acyl-CoA. The dehydrogenases are flavoproteins and contain a tightly bound molecule of flavin adenine dinucleotide, FAD, as the coenzyme. The electrons of the FADH2 via another flavoproteins are transferred to the respiratory chain to give two ATP molecules. The increment 2 double bond formed has a trans-geometrical configuration. The double bonds naturally occurring in fatty acids are in CIase form. E. Enoyl-CoA hydratase hydrates the increment 2 unsaturated acyl-CoA. This enzyme has broad specificity and can act on alpha, beta, or increment 2 unsaturated CoA in trans or CIS configuration. The product formed is L plus beta hydroxyacyl CoA. When the trans double bond is hydrated, the D isomer is formed with CIS double bond. E. Beta hydroxyacyl CoA dehydrogenase oxidizes beta hydroxyacyl CoA by an NAD plus linked reaction that is absolutely specific for L stereoisomer. The electrons from the NAD generated are passed on to NAD dehydrogenase of the respiratory chain, and finally 3 ATP molecules are formed. IV. Acetyl-CoA acyl transferase or thiolase catalyzes a thiolytic cleavage, i.e. involving SH group, and gives acetyl-CoA and acyl-CoA, which is shortened by 2 C atoms. The entire sequence of the oxidation of fatty acid, right from the activation stage to beta oxidation, is given in figure 7.3. The shortened fatty acyl-CoA from one cycle is further oxidized in successive passes until it is entirely converted to acetyl-CoA. It is important to note that a majority of natural lipids contain an even number of carbon atoms. A small proportion that contain odd numbers, upon complete beta oxidation, yield acetyl-CoA units plus a single mole of propionyl-CoA. The propionyl-CoA is converted, in an ATP-dependent pathway, to succinyl-CoA. The succinyl-CoA can then enter the citric acid cycle for further oxidation. Before we conclude, let us look at the energetics of the beta oxidation process, discussed above. Table 7.1 presents the outcome. As is evident, 2 mole equivalents of ATP are used during the activation of the fatty acid. On the other hand, the electrons of the FADH2 are transferred to the respiratory chain to give 2 ATP molecules. The electrons from the NAD generated are passed on to the respiratory chain, and finally 3 ATP molecules are formed. Oxidation of acetyl-CoA gives 12 moles of ATP. Each acetyl-CoA in citric acid cycle gives 3 NADH and 1 FADH2 and 1 GTP for total 12 ATP. Reaction moles of ATP gained: lost activation reaction 2 first dehydrogenation, FAD, plus 2 second dehydrogenation, NAD, plus 3 oxidation of acetyl-CoA 12. Let us understand this by taking an example of palmitic acid, C16. Palmitoyl-CoA will give 8 acetyl-CoA molecules. 1 acetyl-CoA gives 12 ATP, hence 8 molecules will give 96 ATP, and will undergo beta oxidation 7 times, since 2 acetyl-CoA molecules are formed in the last cycle. Activation step is only once. The overall ATP yield therefore is, activation step, 2 times 1 equals 2 beta oxidation. 7 times 5 equals 35 acetyl-CoA. 8 times 12 equals 96, total equals 129. Figure 7.3. Fatty acid activation, transport and beta oxidation. Complete oxidation of one molecule of palmitic acid gives 129 ATP molecules. Similarly, the net result of the oxidation of 1 mole of oleic acid, an 18-carbon fatty acid, will be 146 moles of ATP. 2 mole equivalents are used during the activation of the fatty acid. With our discussion above, we have come to the end of our study on fatty acid oxidation. Next, we shall learn about the oxidation of unsaturated fatty acid. 7.2.2 Oxidation of mono and polyunsaturated fatty acids. The oxidation of unsaturated fatty acids is essentially the same process as for saturated fats, as discussed above, except when a double bond is encountered. In such a case, the bond is isomerized by a specific enoyl-CoA isomerase and oxidation continues. In the case of linoleic acid, the presence of the C12 unsaturation results in the formation of a denoyl-CoA during oxidation. This molecule is the substrate for an additional oxidizing enzyme, the NADPH requiring 2,4-denoyl-CoA reductase. Thus, oxidation of unsaturated fatty acids require increment 3 CIS or trans increment 2 trans-enoyl-CoA isomerase and NADPH-dependent 2,4-denoyl-CoA reductase, in addition to the enzymes of beta oxidation. Hence, these two enzymes are also referred to as additional or auxiliary enzymes. To help you understand the process and the role of the auxiliary enzymes in the oxidation process of unsaturated fatty acids, we have included the oxidation of oleic acid and linoleic acids, two of the unsaturated fatty acids, here in box 1. We have presented these oxidation reactions only to help your understanding on the subject and not primarily for examination purposes. This is extra reading material. Do not get bogged down by these reactions. In fact, you should practice these reactions using linolenic acid, C18, increment 9, 12, 15, and arachidonic acid, C20, increment 5, 8, 11, 14, also. Box 1. Oxidation of unsaturated fatty acids. Oxidation of unsaturated fatty acids given in figures 7.4 and 7.5 are involved herein. In figure 7.4, you can see that oleic acid, C18, has a double bond between carbon 9 and 10. All naturally occurring double bonds are in CIS configuration. Usual 3 turns of beta oxidation results in the formation of 3 molecules of acetyl-CoA, and oleic acid becomes AC12 fatty acid, with the original CIS double bond now being between carbons 3 and 4, i.e. it is increment 3 CIS enoyl-CoA. This is an inactive substrate in beta oxidation. So another enzyme, increment 3 CIS or trans, right pointing arrow, increment 2 trans enoyl-CoA isomerase, converts the CIS double bonds to trans, which is required in beta oxidation, and puts the double bond in positions 2 to 3. Now increment 2 trans enoyl-CoA is formed, and it goes through 5 more turns of beta oxidation, forming 6 acetyl-CoA molecules. Next, let us look at the oxidation of linoleic acid. As evident in figure 7.5, linoleic acid has 18 carbon atoms, with 2 double bonds in positions 9 to 10 and 12 to 13, respectively. It goes through 3 turns of beta oxidation, forming 3 acetyl-CoA molecules and AC12 fatty acid with 2 CIS double bond in 3 to 4 and 6 to 7 positions. This is acted upon by the auxiliary enzyme increment 3 CIS or trans, increment 2 trans enoyl-CoA isomerase, forming increment 2 trans increment 6 CIS denoyl-CoA. Now one more turn of beta oxidation takes place, removing 1 molecule of acetyl-CoA and forming increment 6 CIS enoyl-CoA. This is acted upon by acyl-CoA dehydrogenase, and an extra trans double bond is introduced in 2 to 3 position, forming increment 2 trans increment 4 CIS denoyl-CoA. Now a second auxiliary enzyme, increment 2 trans increment 4 CIS denoyl-CoA reductase, utilizing NADPH, reduces 1 double bond and forms increment 3 trans enoyl-CoA, while the configuration trans of the double bond is suitable for beta oxidation, position 3 to 4 is not suitable. So the first auxiliary enzyme, increment 3 CIS or trans, right pointing arrow, increment 2 trans enoyl-CoA isomerase, changes the position and forms increment 2 trans enoyl-CoA. This goes through 4 more turns of beta oxidation, forming 5 more acetyl-CoA molecules. Thus, with the help of these 2 auxiliary enzymes, linoleic acid can be oxidized completely to 9 molecules of acetyl-CoA. In this way, all unsaturated fatty acids can undergo beta oxidation. So we have seen that the oxidation of fatty acids yields acetyl-CoA, which is a major source of useful metabolic energy. Now, what is the metabolic fate of acetyl-CoA? Read and find out in the next section. But first, let us recapitulate what we have learned so far. Having studied the oxidation of fatty acid, next we shall learn about the synthesis of fatty acids. 7.2.3 Lipogenesis, synthesis of fatty acids. You would notice that acetyl-CoA can react reversibly in the degradation or synthesis of lipids. Above, we saw that fatty acids are degraded to acetyl-CoA. Interestingly, the formation of lipids too starts with acetyl-CoA. Let us see how. When there is an oversupply of dietary carbohydrates, CHOs, the excess CHOs are converted to triacylglycerols. You already know that triacylglycerols constitute molecules of glycerol to which 3 fatty acids have been esterified. In other words, fatty acids are stored primarily in the adipose tissue as triacylglycerol. Further, individuals on low-fat diets also convert glucose to triacylglycerol, which is stored. This involves the synthesis of fatty acids from acetyl-CoA and the esterification of fatty acids in the production of triacylglycerol. The process is called lipogenesis. In fact, the sequence of reactions involved in the formation of lipids is known as lipogenesis. Lipogenesis is not simply the reversal of the fatty acid degradation, but thus starts with acetyl-CoA and does build up by the addition of two carbons units. The fatty acid synthesis occurs in the cytoplasm in contrast to the degradation, oxidation, which you may recall reading earlier, occurs in the mitochondria. The major lipogenic tissues are the intestine, liver and adipose tissue. During lactation, the mammary gland also becomes a major site for lipogenesis and places a heavy demand on a continuing supply of glucose for the synthesis of milk lipids. The metabolic reactions involved in the synthesis of fatty acid are illustrated step by step in Figure 7.6. The synthesis occurs in the cytosol from acetyl-CoA. This is also called de novo, a fresh, synthesis.
When glucose is abundant and the amount of citrate in the mitochondrial matrix exceeds the demand by the citric acid cycle, the excess citrate is transported out of the mitochondria into the cytosol by tricarboxylate translocase. Here, as you can see in Figure 7.6, the citrate is cleaved by citrate lyase to provide the acetyl group for fatty acid synthesis. Besides acetyl-CoA, NADF is also required. The NADF necessary for fatty acid synthesis derives from the conversions of malic enzyme, glucose 6-P dehydrogenase, gluconate 6-P dehydrogenase, and NUC-dependent isocitrate dehydrogenase. The key regulating enzyme of lipogenesis is acetyl-CoA carboxylase. It catalyzes the synthesis of malonyl-CoA from acetyl-CoA and CO2. In the formation of malonyl-CoA via acetyl-CoA carboxylase, biotin is tightly bound to the enzyme as a prosthetic group and acts as a carrier of a carboxyl group that is transferred to acetyl-CoA. The formation of malonyl-CoA signals the beginning of the synthesis of fatty acid. The synthesis of fatty acids from acetyl-CoA and malonyl-CoA is carried out by fatty acid synthase, FAS. Actually, many of the enzymes for the fatty acid synthesis are organized into a multi-enzyme complex called fatty acid synthase. The sequence of reactions catalyzed by this enzyme, as presented in Figure 7.6, can be represented by the following 7 reactions. In the first reaction, acetyl-CoA is added to a cysteine group of the condensing enzyme, CE, domain. Acetyl-CoA plus CECYH, acetyl-CYH CE plus CoASH. Mechanistically, this is a two-step process in which the group is first transferred to the ACP, acyl carrier peptide, and then to the cysteine group of the condensing enzyme. Domain. In the second reaction, malonyl-CoA is added to the ACP sulfhydryl group. Malonyl-CoA plus ACP malonyl ACP plus CoASH. This group is a part of a phosphopentethenic acid prosthetic group of the ACP. In the third reaction, the acetyl group is transferred to the malonyl group with the release of carbon dioxide. Malonyl ACP plus acetyl-CYC beta-ketobutyryl ACP plus CO2. In the fourth reaction, the keto group is reduced to a hydroxyl group by the beta-ectosyl reductase activity. Beta-ketobutyryl ACP plus NADF plus H plus beta-hydroxybutyryl ACP plus NUC plus. In the fifth reaction, the beta-hydroxybutyryl ACP is dehydrated to form a transmonounsaturated fatty acyl group by the beta-hydroxyacyl dehydratase activity. Beta-hydroxybutyryl ACP to butanol ACP plus H2O. In the sixth reaction, the double bond is reduced by NADF, yielding a saturated fatty acyl group to carbons longer than the initial one, and acetyl group was converted to A. Butyl group in this case. To butanol ACP plus NAF plus H plus butyryl ACP plus NUT plus. The butyl group is then transferred from the ACP sulfhydryl group to the CE sulfhydryl. Butyryl ACP plus CECYS ACP plus butyryl CYC. This is catalyzed by the same transferase activity as was used previously for the original acetyl group. The butyl group is now ready to condense with the new malonyl group, third reaction. Above, to repeat the process. When the fatty acyl group becomes 16 carbons long, a thioesterase activity hydrolyzes it, forming free palmitate. Palmitoyl ACP plus H2. O palmitate plus ACP. Go through these reactions carefully. Initially, they might seem a bit tough, but if you follow the sequence as presented in Figure 7.6, you will understand the process of synthesis better. So you notice, the primary fatty acid synthesized by FUS is palmitic acid. Once an acetyl group and a malonyl group are bound to the fatty acid synthase, 7 rounds of enzymatic reactions proceed for the synthesis of palmitic acid, which is then released from the complex. The overall reaction is as follows. Acetyl-CoA plus 7 malonyl-CoA plus 14 NADH plus 14 H plus. Palmitic acid plus 8 CoSH plus 14 NUT plus 6 H2. O plus 7 CO2. Palmitate is then released from the enzyme and can then undergo separate elongation and or unsaturation to yield other fatty acid molecules. We shall learn about this next. So we have seen that in the opposite of fatty acid degradation, which is located within the mitochondria, de novo synthesis of fatty acids takes place within the cytosol. The primary fatty acid synthesized by FUS is palmitic acid. Palmitic acid can be converted to other unsaturated fatty acids. Let us see how. Synthesis of other unsaturated fatty acids. Palmitic acid may be converted to stearic acid, 18O by elongation of the carbon chain. Desaturation of stearic acid produces oleic acid, C18, 19. The enzymes are located in the mitochondria and endoplasmic reticulum and can use fatty acid of varying chain lengths and degrees of unsaturated as substrates. Desaturation occurs in the ER membranes as well, and in mammalian cell S involves 4 broad specificity fatty acyl-CoA desaturases, non-heme iron-containing enzymes.
As we shall encounter later, arachidinate is a precursor for the eicosanoids, the prostaglandins and thromboxanes. It is this role of fatty acids in eicosanoid synthesis that leads to poor growth, wound healing and dermatitis in persons on fat-free diets. Also, linoleic acid is a constituent of epidermal cells sphingolipids that function as the water permeability barrier in the skin. Before we end our study on fatty acid degradation and synthesis, let us recapitulate the salient features of the two processes. Table 7.1 gives the comparison of fatty acid synthesis and degradation. This will help you understand the two processes better. Table 7.1 Comparison of fatty acid synthesis and degradation. Next, we shall focus on a family of compounds called eicosanoids. What are eicosanoids? What is their role in the body? You may recall reading about them in Unit 2 subsection 2.3.5. We suggest you look up this section again now. So then what do you find? Yes, eicosanoids are derived from polyunsaturated fatty acids. We shall learn about their metabolism next. 7.2.4 Metabolism of eicosanoids. You already know now that prostaglandins and the related compounds such as thromboxanes and leukotrienes, collectively known as eicosanoids, are extremely potent compounds that elicit a wide range of physiologic responses. These compounds have extremely short half-life and are produced in very small amounts. They have been compared to hormones in terms of their actions, but they differ from the true hormones in that they are formed in almost all tissues rather than in specialized glands. They generally act locally rather than after transport in the blood to distant sites of action. Prostaglandins are metabolized to inactive products at their site of synthesis and are not stored to any appreciable extent. Let us see how these eicosanoids are synthesized in our body, starting with prostaglandins. Here, we are not going into the details of the reactions involved in the synthesis of these eicosanoids. As dietitians, it is only important for us to understand that eicosanoids are derived from essential fatty acids, EFAs. A. Synthesis of prostaglandins. The dietary precursor of the prostaglandins is the essential fatty acid linoleic acid. It is converted to its immediate precursor of the prostaglandins 20C, PUFA containing 3, 4 or 5 double bonds. Arachidonic acid is the precursor of the predominant classes of prostaglandins. B. Synthesis of leukotrienes. Arachidonic acid is converted to a variety of hydroperoxy acids by a separate pathway involving a family of lipoxygenases. For e.g., neutrophils contain 5 lipoxygenase, which converts arachidonic acid to 5-hydroxy 6, 8, 11, 14 icosahexatetranoic acid, 5-HPETE. 5-HPETE is converted to a series of leukotrienes. 7.3 Lipid Metabolism. Earlier in section 7.2, we got to know about the fate of fatty acids in our body and also how they are synthesized. Now, in this section we shall focus on the metabolism of lipids such as triacylglycerol, cholesterol, phospholipids, etc. You have already learnt about the structure and properties of these compounds in Unit 2, section 2.2 and 2.3. Do look up this unit once again, as it will help you understand their metabolism better. We start with the metabolism of triacylglycerol. 7.3.1 Metabolism of triacylglycerols. The synthesis of triacylglycerol takes place in the endoplasmic reticulum. In liver and adipose tissue, fatty acids in the cytosol obtained from the diet or from de novo synthesis of palmitic acid become inserted into the endoplasmic reticulum, membrane. Fatty acids, you learnt earlier, are stored for future use as triacylglycerols in all cells, but primarily in adipocytes of adipose tissue. Triacylglycerols constitute molecules of glycerol to which 3 fatty acids have been esterified. The fatty acids incorporated into triacylglycerols are activated to acyl cobalt 3 arsenide through the action of acyl-CoA synthetases. This is the same kind of reaction which you have seen earlier in activation of fatty acid prior to oxidation. A membrane-bound acyl-CoA transferase then esterifies 2 molecules of acyl-CoA with glycerol 3-phosphate to form phosphatidic acid, i.e. 1,2-diacylglycerol phosphate. This phosphatidic acid contains glycerol with a fatty acid each in carbon 1 and 2 and phosphate group in carbon 3. The phosphate is then removed by phosphatidic acid phosphatase to yield 1,2-diacylglycerol, the substrate for addition of the third fatty acid. Phosphatidic acid phosphatase releases phosphate and in the membrane, 1,2-diacylglycerol is esterified with a third molecule of fatty acid. In the intestine, triacylglycerol synthesis also occurs in the membrane, but the starting material is 2-monoacylglycerol, which is glycerol esterified with a fatty acid at carbon 2, as can be seen in figure 7.7. In the liver and intestine, triacylglycerol is packaged into lipoproteins, which are then secreted into the circulation. Next, let us learn about the synthesis of phospholipids. 7.3.2 Synthesis of phospholipids. Phospholipids are synthesized by esterification of an alcohol to the phosphate of phosphatidic acid, 1,2-diacylglycerol 3-phosphate. Most phospholipids have a saturated fatty acid on C1 and an unsaturated fatty acid on C2 of the glycerol backbone. The most commonly added alcohols, serine, ethanolamine and choline, also contain nitrogen that may be positively charged, whereas glycerol and inositol do not. The major classifications of phospholipids are phosphatidylcholine, PC, phosphatidylserine, PS, phosphatidylglycerol, PG, phosphatidylethanolamin, PA, and phosphatidylanoacetol, PI. Phosphatidylcholine, a major phospholipid constituent of membranes and lipoproteins, is synthesized de novo in liver cells. The synthesis occurs in the and is linked through 1,2-diacylglycerol with the synthesis of triacylglycerol. Three compounds specifically involved in the synthesis of phosphatidylcholine are A. Choline, B. Choline phosphate, and C. Cytidine diphosphatidylcholine, CDP choline. The synthesis of phosphatidylcholine, with the pathway and the enzymes involved, is given in the figure 7.8. As can be seen, choline is activated first by phosphorylation and then by coupling to CDP prior to attachment to phosphatidic acid. PC is also synthesized by the addition of choline to CDP-activated 1,2-diacylglycerol. Figure 7.8, Synthesis of phosphatidylcholine, PC. A third pathway to PC synthesis involves the conversion of either PS or PA to PC as shown in figure 7.9. The conversion of PS to PC first requires decarboxylation of PS to yield PA. This then undergoes a series of 3 methylation reactions utilizing sadenosylmethionine, SAM, as a methyl group donor. Figure 7.9, Synthesis of phosphatidylserine, PS, and phosphatidylcholine, PC, from phosphethanolamine, PA. Phosphatidylserine arises by an exchange of the ethanolamine residue of phosphatidylethanolamin for a seryl group. Decarboxylation of the serine of phosphatidylserine reforms phosphatidylethanolamin. 3 successive methylation reactions convert phosphatidylethanolamin to phosphatidylcholine. S-adenosylmethionine is the methyl group donor. We move on next to the metabolism of cholesterol. 7.3.3 Metabolism of cholesterol. Cholesterol, as you may already know, is involved in 2 major biological processes. A. It is a structural component of cell membranes, and B. Steroid hormones, vitamin D3, cholecalciferol, and the bile salts are derived from the parent compound. Cholesterol is synthesized de novo in the liver and the intestinal epithelial cells and is also derived from dietary lipids. De novo synthesis of cholesterol is regulated by the amount of cholesterol and triglyceride in the dietary lipid. Let us learn how it is synthesized. A. Cholesterol biosynthesis in liver and intestinal epithelium. The biosynthesis of cholesterol, a complex molecule with 27 carbon atoms, starts with the 2-carbon atom compound acetyl-CoA, which is converted to isopentenyl pyrophosphate, an isoprene derivative with 5 carbon atoms, and then squalene, 30 carbon atoms, which is finally cyclized to cholesterol. It involves 32 different enzymes, some of which are soluble in the cytosol and others of which are bound to the The process has 5 major steps, which are listed herewith. 1. Acetyl-CoA is converted to 3-hydroxy-3-methylglutaryl-CoA, HMG-CoA. 2. HMG-CoA is converted to mevalonate. 3. Mevalonate is converted to the isoprene-based molecule, isopentenyl pyrophosphate, IPP, with the concomitant loss of CO2. 4. IPP is converted to squalene through a series of steps. 5. Squalene is converted to cholesterol. The pathway of cholesterol synthesis is presented in the figure 7.10. For those of you who would want to know a bit more of each of these steps as illustrated in figure 7.10, the following description will be useful. The acetyl-CoA utilized for cholesterol biosynthesis is derived from an oxidation reaction, e.g., fatty acids or pyruvate, in the mitochondria and is transported to the cytoplasm by the same process as that described for fatty acid synthesis. Acetyl-CoA can also be derived from cytoplasmic oxidation of ethanol. All the reduction reactions of cholesterol biosynthesis use NADPH as a cofactor. Acetyl-CoA units are converted to mevalonate by a series of reactions that begins with the formation of HMG-CoA. Unlike the HMG-CoA formed during ketone body synthesis in the mitochondria, this form is synthesized in the cytoplasm. However, the pathway and the necessary enzymes are the same as those in the mitochondria. 2 moles of acetyl-CoA are condensed in a reversal of the thiolase reaction, forming acetoacetyl-CoA. Acetoacetyl-CoA and a third mole of acetyl-CoA are converted to HMG-CoA by the action of HMG-CoA synthase. HMG-CoA is converted to mevalonate by HMG-CoA reductase, HMGR. This enzyme is bound in the endoplasmic reticulum, HMGR absolutely requires NADPH as a cofactor, and 2 moles of NADPH are consumed during the conversion of HMG-CoA to mevalonate. The reaction catalyzed by HMGR is the rate-limiting step of cholesterol biosynthesis, and this enzyme is subject to complex regulatory controls. You will learn more about this later in the section on regulation of cholesterol synthesis. Mevalonate is then activated by 3 successive phosphorylations, yielding 5-pyrophosphomvalonate. In addition to activating mevalonate, the phosphorylations maintain its solubility, since otherwise it is insoluble in water. After phosphorylation, an ATP-dependent decarboxylation yields isopentenyl diphosphate, IDP, an activated isoprenoid molecule. Isopentenyl diphosphate is in equilibrium with its isomer, dimethylallyl diphosphate, DMDP. One molecule of IDP condenses with one molecule, figure 7.10, synthesis of cholesterol of DMDP to generate geranyl diphosphate, GDP. GDP further condenses to combine with another IDP molecule to yield farnesyl diphosphate, FDP. Finally, the NADP-requiring enzyme, squalene synthase, catalyses the head-to-tail condensation of 2 molecules of FDP, yielding squalene. Squalene synthase also is tightly associated with the endoplasmic reticulum. Squalene undergoes a 2-step cyclization to yield lanosterol. By the term cyclization we mean changing an open-chain hydrocarbon to a closed ring. The first reaction is catalyzed by squalene monooxygenase or squalene epoxidase. This enzyme uses NADPH as a cofactor to introduce molecular oxygen as an epoxide at the 2,3 position of squalene. Through a series of 19 additional reactions, lanosterol is converted to cholesterol. B. Regulation of cholesterol synthesis. The cellular supply of cholesterol is maintained at a steady level by 3 distinct mechanisms. Of these 3 mechanisms, regulation of HMG-CoA reductase, HMGR, activity is the primary means for controlling the level of cholesterol biosynthesis. HMG-CoA reductase is an intrinsic membrane protein of the The enzyme's active site extends into the cytosol. HMGR is the rate-limiting enzyme in cholesterol synthesis and is subject to different types of metabolic control, which include A. Feedback inhibition. Cholesterol is a feedback inhibitor of HMG-CoA reductase, thus decreasing further cholesterol synthesis. B. Hormonal regulation. HMG-CoA reductase activity is controlled hormonally. Glucagon favors the formation of the inactive, phosphorylated form of HMG-CoA reductase and hence decreases the rate of cholesterol synthesis. In contrast, insulin favors formation of the active, unphosphorylated form of HMG-CoA reductase and results in an increase in the rate of cholesterol synthesis. C. Sterol-mediated regulation of transcription. The synthesis of cholesterol is also regulated by the amount of cholesterol taken up by the cells during lipoprotein metabolism. Chylomicron remnants internalized by liver cells and LDL internalized by cells of liver and peripheral tissues provide cholesterol, which causes a decrease in de novo cholesterol synthesis. You have already studied about chylomicrons and LDL in Unit 2. Chylomicron and LDL are lipoprotein complexes. You will read in details about these complexes in the next section on lipoprotein synthesis. D. Inhibition by drugs. Lovastatin and mevastatin are reversible competitive inhibitors of HMG-CoA reductase. They are used to decrease plasma cholesterol levels in patients with hypercholesterolemia. Finally, let us look at the degradation of cholesterol in our body. C. Degradation of cholesterol. The ring structure of cholesterol cannot be metabolized to CO2 and H2O in humans. Rather, the intact sterile ring is eliminated from the body by a. Conversion to bile acids which are excreted in the feces, b. Secretion of cholesterol into the bile, which transports it to intestine for elimination is modified by bacteria before excretion, Figure 7.11 summarizes the sources of liver cholesterol and routes by which cholesterol leaves the liver. Figure 7.11 Major routes by which cholesterol leaves liver. 7.3.4 Lipoprotein metabolism. Lipoproteins, as we already know, are the compounds of protein that carry fats and fat-like substances, such as cholesterol, in the blood. You may recall reading earlier about these compounds in Unit 5. The principal lipids carried by lipoprotein particles are triacylglycerols and cholesterol, free or esterified, obtained either from the diet or de novo synthesis. Let us learn a bit more about these plasma proteins. A. Plasma lipoproteins. An introduction. The plasma lipoproteins are molecular complexes of lipids and specific proteins called apolipoproteins. In fact, lipoproteins are composed of a neutral lipid core, containing triacylglycerol in cholesterol esters or both, surrounded by a shell of apolipoproteins, apoproteins, phospholipids and non-esterified cholesterol, all oriented so that their polar portions are exposed on the surface of the lipoprotein. This makes the particle soluble in aqueous medium. What are apolipoproteins? You have seen that various types of proteins are present along with different types of lipids in the lipoproteins. These proteins are specifically referred to as apolipoproteins. You have already come across this concept earlier while studying about enzymes in Unit 4. Do you recall reading about apoproteins? Yes, we learned that many enzymes consist of the protein molecule along with the non-protein molecule. The protein molecule is referred to as apoenzyme. Apolipoprotein and apoenzymes are also called by a general term apoprotein, indicating that a non-protein portion is also associated with it. These dynamic particles, the lipoproteins, are in constant state of synthesis, degradation and removal from the plasma. Lipoproteins function both to keep lipids soluble as they transport them in the plasma, and to provide an efficient mechanism for delivering their lipid contents to the tissues. In humans, the delivery system is less perfect than in other animals, and as a result, humans experience a gradual deposition of lipids, especially cholesterol, in tissues. This is potentially life-threatening occurrence when the lipid deposition contributes to plaque formation, causing the narrowing of blood vessels, a condition known as atherosclerosis, about which you may recall studying in the course Applied Physiology in Unit 4. Do you recall the lipoproteins highlighted in Unit 5? For your information, Table 7.2 here presents the different lipoproteins and their composition. You would notice that the different lipoproteins are classified based on their size and density. Table 7.2 Composition of the plasma lipoprotein %. The chylomicrons are the lipoprotein particles lowest in density and largest in size, and contain the most lipid and the smallest percentage of protein, as can be seen in Table 7.2. Chylomicrons function to deliver dietary triacylglycerols to adipose tissue and muscle and dietary cholesterol to the liver. VLDLs and LDLs are successively denser, having a higher content of protein and a lower content of lipid. HDL particles are the densest of the plasma lipoproteins. The apolipoproteins associated with lipoprotein particles have a number of diverse functions. It, A, serves as structural components of the particles, B, provides recognition sites for cell surface receptors, and C, serves as activators or coenzymes for enzymes involved in lipoprotein metabolism. Apolipoproteins are divided by structure and function into classes A to H, with most classes having subclasses, for example, ApoAI and ApoC2. Having got a basic insight into the structure, composition of lipoproteins, we shall now move on to read about the metabolism of these compounds, starting with chylomicrons. B. Metabolism of chylomicrons. Chylomicrons are assembled in intestinal mucosal cells and carry dietary triacylglycerol, cholesterol and cholesterol esters, plus additional lipids made in these cells, to the peripheral tissues. The particle released by the intestinal mucosal cell is called a nascent chylomicron and contains apolipoprotein B48, ApoB48, as shown in the figure 7.12. Chylomicrons leave the intestine via the lymphatic system and enter the blood circulation. When it reaches the plasma, the nascent chylomicron is rapidly modified, receiving ApoE and ApoC apolipoproteins from plasma HDLs, which is required for the activation of lipoprotein lipase. Figure 7.12 Metabolism of chylomicrons in the capillaries of adipose tissue and muscle. The fatty acids of chylomicrons are removed from the triacylglycerols by the action of lipoprotein lipase, LPL, which is found on the surface of the endothelial cells of the capillaries. Lipoprotein lipase is an extracellular enzyme that hydrolyzes triacylglycerol into 2-monoacylglycerol and 2 fatty acids as indicated in the figure 7.12. The ApoC2 in the chylomicrons activates LPL in the presence of phospholipid. The free fatty acids then enter the cells passively down a concentration gradient, and the glycerol backbone of the triacylglycerols is returned via the blood to the liver and kidneys. Patients with a deficiency of lipoprotein lipase or ApoC2 show a dramatic accumulation of triacylglycerol-rich lipoproteins in the plasma, for example, type 1 hyperlipidemia, familial hyperchylomicronemia. As the chylomicron circulates and the triacylglycerol in its core is degraded by lipoprotein lipase, the particle decreases in size and increases in density, since it has lost a considerable amount of its lipid component. In addition, the C apoproteins are returned to the HDLs, from which they were originally obtained. The remaining particle left is called a remnant. Chylomicron remnants containing primarily cholesterol, ApoE and ApoB48 are then delivered to and taken up by the liver through interaction with the chylomicron remnant receptor. The recognition of chylomicron remnants by the hepatic remnant receptor requires ApoE. Chylomicron remnants bind to these receptors and are taken into the cells by endocytosis. The endocytosed vesicle then fuses with a lysosome, and the apolipoproteins, cholesterol esters, and other components of the remnant are hydrolytically degraded, releasing amino acids, free cholesterol and fatty acids. The cholesterol released from the chylomicron regulates the rate of de novo cholesterol synthesis in the liver by causing a decrease in cell content of HMG-CoA reductase, which you learned earlier is the key enzyme in cholesterol synthesis, as well as by inhibiting the enzyme. C. Metabolism of very low-density lipoproteins, VLDL. VLDLs are produced in the liver. They are composed predominantly of triacylglycerols, TG, cholesterol and cholesterol esters, C, and their function is to carry this lipid from the liver to the peripheral tissues. There, the triacylglycerol is degraded by lipoprotein lipase, as discussed, for chylomicron degradation. The process involved thereafter is illustrated in figure 7.13, and the process includes 1. Release of VLDL. VLDLs are released from the liver as nascent VLDL particles containing apolipoproteins B100 and AI. They must obtain ApoC2 and ApoE from circulating HDL as shown in figure 7.13. As with chylomicrons, ApoC2 is required for activation of lipoprotein lipase. 2. Modification of circulating VLDL. Next, as VLDLs pass through the circulation, their structure is altered. Fatty acids and glycerol are removed by lipoprotein lipase, causing the VLDL to decrease in size and become denser to form intermediate density lipoproteins, IDL. Surface components, including the C and E apolipoproteins, are transferred to HDL. Finally, cholesterol esters are transferred from HDL to VLDL in an exchange reaction that concomitantly transfers triacylglycerol or phospholipid from VLDL to the HDL. This exchange is accomplished by cholesterol ester transfer protein. 3. Production of LDL from VLDL in plasma. After these modifications, the VLDL has been converted in the plasma to LDL. An intermediate-sized particle, the intermediate density lipoprotein, IDL, as shown in figure 7.13, is observed during the transition from VLDL to LDL in the plasma. IDLs can also be taken up by cells through receptor-mediated endocytosis. Figure 7.13, Metabolism of VLDL. Next, let us look at the metabolism of LDL. D. Metabolism of low-density lipoproteins, LDL. LDL, as seen earlier, contains much less triacylglycerol than its VLDL predecessors and has a high concentration of cholesterol and cholesterol esters. The primary function of LDL particles is to provide cholesterol to the peripheral tissues. How do they do that? In fact, a multi-step process is involved in the metabolism of LDL. We shall not go into the details of these steps, but certainly look at the mechanism involved in simple terms. LDL particles provide cholesterol to the peripheral tissues by depositing free cholesterol on the membranes of cells as they come in contact with the cell surface and by binding to receptors on cell surface membranes that recognize apolipoprotein B100. LDL receptors are negatively charged glycoprotein molecules that are clustered in pits on cell membranes. The intracellular side of the pit is coated with the protein clathrin, which stabilizes the shape of the pit. After binding, the LDL is internalized as intact particles by endocytosis. The vesicle containing the LDL rapidly loses its clathrin coat and fuses with other similar vesicles, forming larger vesicles called endosomes. The pH of the contents of the endosome falls, allowing separation of the LDL from its receptor. The receptors then migrate to one side of the endosome, whereas the LDL stay free within the lumen of the vesicle. The receptors can be recycled, whereas the lipoprotein remnants in the vesicle are degraded by lysosomal, hydrolytic enzymes, releasing cholesterol, amino acids, fatty acids and phospholipids. These compounds can be recycled by the cell. The number of receptors for lipoproteins vary according to the availability of these lipoprotein particles and according to the needs of the cell. For example, if there is a large amount of a particular circulating plasma lipoprotein, the number of cell surface receptors for it decreases, frequently termed downregulation. Conversely, if cells are starved for cholesterol, they increase the number of cell surface receptors, i.e., upregulation. Lastly, we come to the metabolism of HDL. Metabolism of high-density lipoproteins, HDL. HDL particles are synthesized in the liver and are released into the bloodstream by exocytosis. HDL performs a number of important functions, as you may have realized while reading through the earlier sections. These include HDL serves as a circulating reservoir of apolipoprotein, ApoC2, the apolipoprotein that is transferred to VLDL and chylomicrons. B. It is an activator of lipoprotein lipase, removing free, unesterified cholesterol from extrahepatic tissues and esterifying it, using the plasma enzyme phosphatidylcholine cholesterol acyltransferase, PCAT, also known as LCAT, where L stands for lecithin. C. It transfers cholesterol esters to VLDL and LDL in exchange for triacylglycerol, and D. It carries cholesterol esters to the liver, where the HDL is degraded and cholesterol is released. A brief discussion of the functions and metabolism of HDL follows. The metabolism of HDL is illustrated in Figure 7.14. Figure 7.14, Metabolism of HDL1. HDL as a reservoir of apolipoproteins. HDL particles not only serve as the source of apolipoproteins required for the proper metabolism of other plasma lipoproteins, but also take back most of these proteins before the chylomicron remnants and LDLs bind to their cell surface receptors and are endocytosed. 2. HDL uptake of free cholesterol. Newly secreted HDL are disc-shaped particles as shown in the figure 7.14, containing predominantly unesterified cholesterol, phospholipids and a number of apolipoproteins including ApoE, ApoA and ApoC. They are rapidly converted to spherical particles as they accumulate cholesterol. HDL particles are excellent acceptors of unesterified cholesterol from the surface of cell membranes and from other circulating lipoproteins. 3. Esterification of free cholesterol. Once free cholesterol is taken up by the HDL, it is immediately esterified by phosphatidylcholesterol acyltransferase, PCAT, a plasma enzyme synthesized by the liver, which is activated by ApoAI of the HDL. Plasma levels of ApoAI are increased by modest alcohol intake. The fatty acid from carbon-2 of phosphatidylcholine is transferred directly to the cholesterol, leaving lysophosphatidylcholine. The resulting cholesterol ester is so hydrophobic that it is effectively trapped in the HDL and can no longer be transferred to a membrane. The only mechanism for removing it from HDL in the plasma is through transfer to VLDL by the cholesterol ester transfer protein, where it ultimately remains in the LDL until that particle is taken up by a cell. About 2/3 of the cholesterol in the plasma is esterified with fatty acid. In liver disease, a decreased concentration of plasma cholesterol esters is observed. This may be due to either a deficiency in phosphatidylcholine production or a lack of PCAT. 4. Fate of HDLs. Spherical HDL particles are taken up by the liver by receptor-mediated endocytosis and the cholesterol esters are degraded. The cholesterol thus released by the action of enzyme hepatic lipase can be repackaged in lipoproteins, converted into bile acids, or secreted into the bile for removal from the body. With the metabolism of HDL, we come to the end of our discussion on metabolism of lipoproteins. We have seen in this section the fate of the different lipoproteins. Serum lipoprotein levels are maintained in the body. What happens when the levels of these lipoproteins are elevated in the body? Read the next section and find out. 7.4 Hyperlipoprotein EMIAS. The term hyperlipoproteinemia describes a group of disorders in which serum lipoprotein levels are elevated. These disorders are classified into 6 types, depending on which lipoproteins are abnormally elevated and are summarized in Table 7.3. Each hyperlipoproteinemia is not a single disease but a group of disorders marked by the same lipoprotein abnormality, and each includes some primary, genetically transmitted, disorders and some secondary disorders. When a disorder of lipid metabolism occurs secondary to a particular disease, e.g. type 4 hyperlipoproteinemia secondary to uncontrolled diabetes, treatment of the underlying illness will frequently correct the lipid abnormality. Similarly, when a primary lipid disorder is aggravated by exogenous obesity, alcohol or glucocorticoids, the elimination of the aggravating factor will make diet therapy easier. Diet also affects the development of hyperlipoproteinemias. The dietary factors causing an increase in plasma lipoproteins in a great many people are obesity and high intake of foods rich in cholesterol and saturated fats. Table 7.3. Types of hyperlipoproteinemia. A brief review follows. A. Type 1 hyperlipoproteinemia is an uncommon pattern marked by elevated chylomicrons. Cholesterol is normal and triglycerides are markedly elevated, usually greater than 1000 mg per deciliter. Among the primary disorders producing this pattern are familial lipoprotein lipase deficiency and apolipoprotein C2 deficiency. B. Type 2 A hyperlipoproteinemia is marked by high LDL with normal VLDL. Plasma cholesterol levels are high but triglycerides are normal. The genetic disorder associated with this pattern is familial hypercholesterolemia, in which there is an autosomal dominant pattern of inheritance. The biochemical defect is a deficiency of LDL receptors. This pattern is also seen secondary to nephrotic syndrome, Cushing's syndrome and hypothyroidism. C. Type 2b hyperlipoproteinemia is a common pattern characterized by increases in VLDL and LDL. Both cholesterol and triglyceride levels are elevated. Familial combined hyperlipoproteinemia, also called familial multiple lipoprotein type hyperlipidemia, is a disorder in which individuals with type 2a, type 2b and type 4 hyperlipoproteinemias are found in the same family. Type E hyperlipoproteinemia can be seen secondary to nephrotic syndrome, Cushing's disease and hypothyroidism. Primary type 2b hyperlipoproteinemia can be aggravated by exogenous obesity or glucocorticoids. D. Type 3 hyperlipoproteinemia is marked by a reduced electrophoretic mobility of VLDL. In this, cholesterol and triglycerides are both elevated, frequently to about the same level. For example, cholesterol and triglycerides may both be 400 mg per deciliter. The primary form of this disorder is called familial dysbeta-lipoproteinemia. These patients accumulate a partially degraded VLDL, beta VLDL. E. Type 4, a common pattern of hyperlipoproteinemia, is marked by elevated VLDL with high cholesterol and triglycerides. 1. The primary disorders associated with this pattern are familial multiple lipoprotein type hyperlipidemia, and the mild form of familial hypertriglyceridemia. 2. In other associated disorders, elevated VLDL is common secondary to diabetes and uremia, and is also associated with hypopituitarism and nephrotic syndrome. Alcohol, glucocorticoids, estrogens and exogenous obesity may aggravate an already elevated VLDL in patients with primary hyperlipidemia, but they seldom induce hyperlipidemia in normal individuals. F. Type 5, a rare pattern, is marked by elevated chylomicrons and VLDL. Both cholesterol and triglycerides are high. 1. The primary disorders with the pattern are familial lipoprotein lipase deficiency, apolipoprotein C2 deficiency, and the more severe form of the familial hypertriglyceridemias. 2. Type 5 hyperlipoproteinemia may be seen secondary to the same disorders as type 4. It is most commonly seen secondary to poorly controlled diabetes. Our reading on this topic would not be complete without some information about the diagnosis of these disorders. We shall learn about this next. Diagnosis of hyperlipoproteinemia. We have already seen how hyperlipoproteinemia is classified based on the elevated levels of lipoproteins such as chylomicron, LDL or VLDL etc. Next, we shall learn why the diagnosis of hyperlipoproteinemia is important and when to do it. Some information of the diagnosis mechanism is also included. 1. Because of the high association of hyperlipidemia with coronary heart disease, it is generally recommended that serum cholesterol and triglycerides be measured periodically, especially in young adults. If there is a history of hyperlipoproteinemia or premature coronary artery disease in the family, children should be tested as well. A. Timing of measurement. The serum cholesterol level is relatively unaffected by eating, but a recent meal can cause marked elevation of triglycerides. Triglycerides should only be measured after a 12 to 14-hour fast. Serum lipids are best determined when the patient is maintaining a steady weight and has been on his usual diet for several weeks. B. Repeat measurements. Before making a firm diagnosis, fasting lipids should be measured 2 or 3 times at 2 to 3 week intervals. 2. The presence of chylomicrons can be determined by refrigerating the plasma at 4 degrees Celsius overnight. If chylomicrons are present, they will form a creamy layer on top of the plasma. The presence of chylomicrons in plasma drawn after a 12-hour fast is indicative of type 1 or type 5 hyperlipoproteinemia. Fasting chylomicronemia is usually seen only with fasting triglyceride levels of greater than 1000 mg per deciliter. 3. The implications of elevated serum cholesterol depend on the lipoprotein with which it is associated. As noted above, the risk of coronary artery disease is highly associated with elevated LDL cholesterol. A marked increase in VLDL may result in some increase in cholesterol in addition to an increase in triglyceride. VLDL contains about 1 mg of cholesterol for every 4 mg of triglyceride. HDL cholesterol is easily determined by precipitating LDL and VLDL with phosphatidylstic acid and magnesium chloride. The non-precipitated cholesterol is HDL. LDL cholesterol can then be calculated. LDL cholesterol equals total cholesterol minus HDL cholesterol minus triglycerides upon 5/4. Most patients with hyperlipidemia can be assigned to a specific hyperlipoproteinemia on the basis of total cholesterol, triglycerides, HDL cholesterol and the refrigerator test for chylomicrons. Isoelectric focusing of apolipoproteins is necessary to establish the diagnosis of key apolipoprotein deficiency and familial dysbeta-lipoproteinemia, type 3 hyperlipoproteinemia. Finally, we shall end our discussion on lipid metabolism by learning about ketosis. What is ketosis? Ketosis is the body's survival system. Let's get to know about this system in our body. 7.5 Ketosis. Being in ketosis means our body has burned a large amount of fat in response to the fact that it did not have sufficient glucose available for energy needs. Under everyday conditions, the carbohydrates we eat are converted to glucose, which you already know is the body's primary source of energy. Whenever our intake of carbohydrates is limited, for a long enough period of time, we will reach a point where our body draws on its alternate energy system, i.e. the fat stores for fuel. The condition called ketosis means our body burns fat and turns it into a source of fuel called ketone bodies. Ketone bodies are produced whenever body fat is burned. When you burn a larger amount of fat that is immediately needed for energy, the excess ketone bodies are discarded in the urine. Let us next see how ketone bodies are formed. Acetyl-CoA is oxidized to CO2 via citric acid cycle, as given in carbohydrate metabolism. Only in liver mitochondria, the acetyl-CoA can be converted to ketone bodies, i.e. acetoacetate, acetone and 3-hydroxybutyrate. The ketone bodies are water-soluble lipid fuels that are continuously released from the liver. When carbohydrate is plentiful and glucose is readily available to the tissues, the amount of circulating ketone bodies is low. When large amounts of triacylglycerols are being hydrolyzed in adipose tissue, in response to an increase in whole body energy demand, the rate of oxidation of fatty acid increases in the liver and other tissues. In the liver, these increases ketogenesis and thus increases the ketone body concentration in the circulation. Normally, some acetoacetate is converted to 3-hydroxybutyrate. Further, acetoacetate and hydroxybutyrate are valuable fuels for skeletal and cardiac muscle. It is estimated that they supply 10% of the daily energy requirement of these tissues. The breakdown of ketone bodies by the peripheral tissues is called ketolysis. When the process of ketogenesis exceeds ketolysis, ketosis or ketoacidosis occurs. Here we must differentiate ketosis from ketoacidosis. Ketosis, we have seen, is our body's natural survival system. Ketoacidosis, on the other hand, is a life-threatening condition most often associated with uncontrolled insulin-deficient type 1 diabetes. In type 1 diabetes, the absence of insulin leads to a toxic buildup of blood glucose and an extreme breakdown of fat and muscle tissue. The presence of insulin keeps ketone bodies' production in check so that a mild, beneficial ketosis is achieved. 7.6 Summary. In this unit, lipid metabolism at cellular level has been discussed at length. First, we have seen how fatty acids are used for the production of energy. Secondly, the body has the capacity to synthesize a variety of fatty acids except the essential fatty acid. Higher fatty acids, i.e. eicosanoids, have important biological functions. The significance of cholesterol in biologic processes along with their synthesis and regulation has been looked into. Lastly, we have seen the transport of lipoproteins and its relation to hypolipoproteinemia. 6 types of hypolipoproteinemia are present. Ketosis occurs when the process of ketogenesis exceeds the process of ketolysis. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.